So let me just start by saying hello to everyone. My name is Rasmus Bro from University of Copenhagen. And today we're going to hear about something that I'm very interested in, about a database called ChemTastesDB, or DB, which Christian Noyes and Davide Bellapio is going to talk about and explain all the details of. And if you have questions, just put them in the chat and I will convey them whenever suitable. So with that, please have a go.
Hello, everyone, and welcome to this presentation. Thank you, Rasmus, for inviting us to be part of this webinar series. Today, David and I are going to present our results in the field of taste chemistry. I'm going to start by presenting the structure of the ChemTastesDB. Only a slide like an introduction. In the taste chemistry, molecules are classified into 5 basic tastes, that is sweet, bitter, umami, sour, and salty. However, there are also molecules that exhibit non-basic tastes. For instance, burning, astringent, pungent, as well as other molecules with exhibit multiple tastes at the same time, which are labeled as multi-taste. Among the basic tastes, sweet, bitter, and umami are receptor-mediated tastes. It means that they are associated with specific receptors located on the tongue. On the other hand, sour is the result of activation of proton-sensitive ion channels, while saltiness is an stimulus produced by soluble salts. Okay, for the generation of the database, we started by using the information available in 88 papers, 3 books, and 54 book chapters. Using all these references, a total of 6,930 compounds were retrieved. All these molecules were designed and optimized in the HyperChem software. The optimization process was performed using molecular mechanics. For those molecules, a chemical name or a CAS registry number, verification process of the chemical structure was conducted in the packet open library. Then, for filtering the dataset, a 9 workflow was programmed to use 5 basic steps to merge the molecules using the name, the CAS registry number, or the packet seed. Then a further matching process was performed by the canonical smiles in order to identify similar compounds. Then molecules were assigned to a single class according to the majority voting approach, while the miscellaneous class was created by including molecules that exhibit non-basic taste, for instance, astringent, cooling, at burning, heating, pungent, or tingling. All these procedures implemented in 9 allowed us to identify 4,075 molecular tastants. However, since some molecules don't have a chemical name, we use a tool implemented in the ALBA molecule software to find the information in the PubChem library. When this option failed, the naming tool as implemented in Marvin Sketch was used to create or to generate the UPAC name. In this way, we created a database that contained 10 classes. 5 classes for the basic taste, that is sweet, bitter, umami, sour, and salty. 5 other classes related to non-basic taste, which are related to non-sweet, non-bitter, tasteless, multi-taste, and miscellaneous classes. The information of the ChemTastesDB is freely available in the Zenodo repository. The second version of the ChemTastesDB was launched on this March 2005. This slide, you can see a code, a QR code to see access to the dataset. Further information of all the workflow, on all the procedures to create the database is available in the we publish in Food Chemistry Molecular Science. So let's take a look at all the files available in the Zenodo repository. First, there is the database file, which is an Excel file containing all the information of each molecular entry. In this particular case, the chemical name, the PubChem seed, the CAS registry number, canonical smiles, the class days, and the reference in which those molecules are available. And the molecules RAR file contains the 4075 hyperchem files, which are useful to calculate or generate molecular descriptor to calibrate the QSAR models. The README file is a PDF file containing all the information about the database. And finally, the references contains all the information related to the sources we use to create dataset. In a further step in this study, we use the molecular tastants available in ChemTastesDB to construct the chemical space. To this end, we calculate a set of 166 molecular access systems, which are molecular descriptors related to the presence or absence of molecular fragments in the chemical structure of tastants. Then the Jakarta-Nimoto binary distance was implemented with the T-distributed stochastic neighbor embedding approach for data reduction in order to visualize in a dimensional map the chemical space of the ChemTastesDB. In this slide, you can see the chemical space created by the projection of the first two dimensions of the TSNE approach. As you can see, there are some molecules overlapping, as well as some classes overlapping. To better understand the nature of the distribution of these molecules in the chemical space, we analyzed a class, non-class plot for the sweet, bitter, umami, and sour taste. In this slide, it's easy to understand or to identify some consistent cluster related to family of compounds of sweeteners, of bitterants, of uminess and sourness molecules. Further information, if you are interested to see the nature of this cluster, please refer to the published work in the Food Chemistry Molecular Science. Okay, what did we do in between the ChemTastesDB and ChemTastesPredictor? Before creating the QSAR models, the classification models for taste predictor, perform a systematic review, which we were able to identify 52 published studies which cover a period of time starting by the first model in 1980, the last works published in 2022. In 1918 appeared the first model for sweet and bitter discrimination. And during this systematic review, we identified 12 models by 2022. 1 year later, in 1981, appeared the first model for sweetness discrimination. In this case, the classification was performed between sweet molecules and non-sweet molecules. It means other tastes not related to the sweeteners. Several years later, in 2006, was published the first model for bitterness prediction, with a total of 14 models published by 2022. In recent years, particularly in 2020, it has been published the first work for umami prediction, and during this systematic review, we identified only one model for sourness prediction, probably because sour compounds are not numerous like sweeteners and bitters. Finally, in 2022, two new models were published, but in this case, not related to binary classification. Those models are related to the discrimination between bitter, sweet, and umami molecules. Okay, now Davide is going to present the results of the ChemTaste predictor, focus on the calibration of machine learning models to predict the taste of molecules included in the ChemTastesDB.
Okay, so I hope you can hear me. Thank you, Christian. Thank you, Christian, for the presentation you did. So
let me stop. Let me
yeah.
stop the presentation. Okay, go ahead, Davide. Thank you.
Okay, so I think now you can see my screen. Okay.
Yes.
Okay. So, so as Christian explained it before, I mean, all the information we let's say organized and structured in the ChemTastesDB. I mean, it's nice because it's the beginning of the modeling that you can do on the basis of let's say the QSAR approach. Okay, so what does it mean? I mean, in the in the ChemTastesDB, you have all the information related to the structure of molecules, and then the labels associated to their taste. Okay, so you can use that information to, let's say, develop by means of machine learning and chemometrics some models which are able to establish a relationship between the molecular structure and the taste. Okay, and these models are predictive. So then, let's say you have a new chemical compound, you don't know the taste of this chemical compound. Well, you can use these models. Okay, you can use as input of these models the structure of the chemical compound, and you can predict the taste. Okay, without any kind of experimental setup at the beginning. Okay, so which is the idea? I mean, this is let's say an old idea. Okay, so QSPR stands for quantitative structure property relationship. Okay, and so again, the idea is to establish a relationship between the structure of chemicals and some properties, okay, which in this case are the taste of chemicals. In order to do that, what do we need? Well, we need a list of compounds, okay, which we use as training data, okay, to develop these models. These models are completely empirical. They are based on machine learning. So we use machine learning to learn, okay, from these training compounds, which can be the relationship between the structure of the chemical and the taste. Okay, so we need a set of training compounds. We have to know the structure, the information on the structure, on the chemical structure, and then of course, I mean, these models are supervised, so we need to know also the label. Okay, so for each chemical, we have to know the experimental label, like sweet and non-sweet, bitter, or whatever. Again, another thing we need is to encode the structure, the chemical structure of the molecules in, let's say, useful numbers, okay, which are called molecular descriptors. So in few words, there are softwares that can be used to establish, to calculate molecular descriptors, which are quantitative numbers, which represent the molecular structure of a compound. And of course, you can calculate many molecular descriptors, and each descriptors can encode a different chemical feature. Okay, so then we have a multivariate modeling because we describe the chemical structure with let's say a vector of numbers. Okay, and then if we collect many molecules, we can get a data set. Okay, and then by means of machine learning and chemometrics, okay, then we can establish a relationship. Now, this is just an example, okay. This is a linear model, let's say. A, B, and C are three molecular descriptors. I mean, it can be, I don't know, the molecular weight, the number of double bonds you have in a molecule, and so on, okay? Just as an example. So these descriptors can be easily calculated by a software in a few seconds. Taste is the taste we want to predict, okay? So if we are lucky, maybe we can establish, as in this slide, a linear relationship between the property, the taste, and our molecular descriptors. And then we get a new molecule. We can get A, B, and C, the molecular descriptors, and we can predict the taste for this new molecule, okay? So this is the idea of QSAR or QSPR, as you want to call these methods, okay? So quantitative structure, activity, or property relationship. So finally, you have you get a predictive model to predict the taste of a molecule on the basis of the molecular structure, which is encoded in some numbers, which are the molecular descriptors. So we have used the database that Christian explained, because in the database you have the information you need to get these models. Okay, so here, for example, is what I was talking about before. Okay, you can organize this information in a data matrix, as we usually do in chemometrics. Or machine learning, or whatever you want to call it. So you have a data matrix where rows represent the molecules, okay? The columns represent the molecular descriptors. So the columns are the features, the variables that we use to describe the molecular structure of all the molecules of the chemicals. And then we have a property, a response, which is in this case, qualitative response, so it's the taste label, okay? So for example, if you want to discriminate sweet from non-sweet chemicals, okay, the taste label will be a binary label, okay, stating if each chemical is sweet or not sweet, okay? And this is actually, as you probably know, the basis for doing machine learning or chemometrics. In this special case, this is the starting point for doing classification. So to establish a model that is able then to predict, and for a new chemical, the class, so the test label. So in the previous example, to predict if a new chemical is sweet or not sweet, umami or not umami, bitter or non-bitter, and so on. Okay. So we did all these models on the basis of the information we had in the database. We did a lot of trials because we had a lot of nice data, but I mean, these are the data that are available and then can be downloaded for free. So we now, when you get molecular descriptors, you can actually calculate different types of molecular descriptors. And if you are not so familiar in this framework, I can maybe give you some references to how to get these molecular descriptors, but it's something easy to get, okay? There are online tools that can be used. So how do you get molecular descriptors? Where usually you encode, as Christian explained it before, you encode the structural information on the molecular structure in a file, okay? And then you use that file as input in a software to get the descriptors. And usually you can get different types of descriptors. So here we tried with some descriptors which are binary descriptors, and they are called fingerprints, and some, let's say, classical molecular descriptors. And then we use different methods for classification. So probably you can, if you are a little bit familiar with chemometrics, you can recognize some of the most, let's say, classical classification approaches: KNN, PLS-DRA, random forest, ADABUS. So we tried different approaches to see on the basis if the classification task was easy or not. So if we could separate classes with a linear approach or not. And we try to model different tasks, let's say. Okay, so for example, to separate sweet and non-sweet chemicals, umami and non-umami, sweet and bitter, and so on. And the results actually were quite good. Of course, we did a lot of validation. So every time we did, let's say, a model, we divided the set of chemicals in a training set and in a test set, and then we used only the training set, let's say, to optimize our model, to select the optimal set of molecular descriptors, to tune all the parameters of the classification models, and then we predicted the test set, okay? Here you can see some of the results, but again, this can be found in our publication. I will give you the link later. So this is the number of, and train and test is the number of molecules that we use to train or to test a model. Okay, so you can see, I mean, the number is enough, I mean, let's say significant, okay? And NER means non-error rate. So it's let's say it's the balanced accuracy. Okay, so let's say it's the percentage of correct prediction. Okay, weighted by the class balance. And so you can see, I mean, all the models can, many of the models we can get very nice accuracy. Okay, so these models are actually able. They are not perfect as usual, but they are able, in most of the cases, to predict the correct taste of chemicals. Yes. So just to conclude, okay, as previously Christian said, the taste prediction is is very important. Even when we did the review of all the models that have been developed in this framework, we could see that there is an increasing use and attention in this field. Even because it's somehow cheap, because once you have some experimental data, then you can just use computational models to get new predictions. Okay. Then in this framework, okay, so the idea of the ChemTastesDB is to give to the scientific community an available and accessible database where scientists can retrieve the information on these chemicals. And of course, this information then can be used in the case to eventually, let's say, develop QSPR models for the prediction of molecular taste. Of course, the database is updated, okay, time to time. So now, as Christian said before, we released the latest version a few months ago. So probably now for some months, the database will be that one, the one you can find online. But of course, in the future, we will update and expand the database. Okay, these are the contacts for me and Christian. And of course, you can contact us if you are then interested in this work or whatever. And this is again the link, okay, of the database, which is accessible in Zenodo. But if you lose the link, then you can write us, and we can provide you with a link afterwards. And I think this is all. So thanks for the attention.
Thank you very much. And if you have questions, feel free to either open your microphone or write it in the chat. Can I ask you, do the chemicals in the database span, are there some deficiencies in what chemicals you have available?
Which you mean which kind of which families of chemicals are available in the database?
Yeah.
I think Christian is the one who can reply to this better than me.
Okay, in the references, there are a lot of chemicals. For example, sucrose, sucrose derivative, isovanillyl derivative, peptides, several families of compounds which were tested for identifying the basic taste or non-basic taste. We use this information, construct the database, and to merge the molecules with similar chemical structure and different taste or similar taste. So we don't focus only on one type of family compound. We use every type of molecules available in the literature.
Mm-hmm. How about, for example, because I was thinking that this could be very interesting for pharmaceutical companies. They would like to predict the taste of APIs that they want to test. So for example, pharmaceuticals, would you, I mean, like relatively small molecules?
I think there are some probably small molecules in the database. One important thing is, I mean, this is, as you know, Rasmus, I mean, it's true in QSPR, but when you use machine learning or chemometrics, of course, when you use these models, this predictive model, then you have always to check for the applicability domain. Okay, so and this is, I mean, better explained in the publication we did. So of course, the prediction is reliable only if the molecule, the target molecule you predict is enough similar to the training molecules. Okay. So of course, I mean, if you predict something which is extremely different as a molecule, then the prediction is not reliable. Okay. I mean, all the methods we use are discriminant methods. So you get a prediction, but then you always have to check for the applicability domain. So how much a molecule is enough similar to the training compounds. Yeah.
Makes sense. Makes sense. There's a question in the chat from JGR. Are you there's several questions. Are you planning to look at compositions of several molecules?
This means mixtures, I guess.
Yeah.
It would be nice. It's a mess. I mean, it's not a mess, but this is something that in QSAR, generally speaking, is a new framework where you can use QSAR. It would be nice, but you need data. So you need, I mean, we got a lot of data specifically for single molecules. Then if you want to look at mixtures, you need some data. And then you have to model the data in a different way, because then you have to somehow represent the mixtures with some descriptors, and you can do it, but it's not so easy. I don't know, Christian, if you have available some data on mixtures of compounds.
Actually, during the systematic review, we identified only 10 experimental data reporting the sweeteners of some mixtures. That's all in the literature.
Okay. Okay.
It's not available enough information in order to perform, for example, QSPR models. Yeah, it's limited the information.
Okay, there's a follow-up question from the same person. Do you predict an intensity of the particular attribute or only its presence?
Yeah, we predict it's qualitative. So we predict, I mean, not the presence, but the class. So, I mean, all these molecules have been labeled, experimentally labeled as sweet, or I don't know, bitter, or sour, or umami. And so we predict if it's sweet or not, if it's sour or not.
Yes.
In addition, complementing the explanation of Davide, in some sources of the database of the ChemTaste database, there are information for predicting sweetness intensity, bitterness intensity, and a few molecules for umami intensity. So in this case, also the intensity information is available. For umami, less information available, but for sweet and bitter, there are plenty of information available in the different sources you can access in the Zenodo repository.
Hi. Yeah, Adnan.
I do have a question. Thank you for the presentation, actually. Just for the stereochemistry, did you take it into account? Because we know that, for example, two different stereochemists, like stereoisomers, like R and S, will have different tastes. Taking example, limonene, the R is sweet and better than me, and the S is bitter. So did you take into account the stereochemistry? Because we know the molecular descriptors, normal molecular descriptor, we need to take into account this for QSAR models, QSPR models. So in fingerprints, did you take it into account?
Oh, it got silent.
Do you want?
No. I understand very well. Don't worry. In the data set, stereochemistry is clearly differentiated. But for calibrating the QSPR model, we only use conformational independent molecular descriptors. So those molecules, ambiguous molecules, were excluded for the modeling process. Yeah. So for using 3D descriptors, we are planning to optimize molecules according to a quantum mechanic procedure in order to 3D descriptor and to discriminate between stereoisomers.
Yeah, if I can add something, because I mean, this is also part of the QSPR, let's say, workflow. So when you use three-dimensional descriptors, then it's not so easy to distribute models. While if you use two-dimensional descriptors, it's more easy because the input that you can use for a new prediction is more easy to get. So we started from the easiest way. Then of course we can improve it. Yeah.
Right. Just I want to add that there are some descriptors or fingerprints that you can use, like the map chiral is this. I don't know the author or developer, but maybe they can share it with you later, and you can use these fingerprints to take into account stereochemistry.
Yeah, sure. If you can share it, it would be nice to add it as a part of the descriptors. Yeah, sure.
And for the balancing the data for the training set, did you take into account, because we know that you have maybe more like sweet or more beta compounds. Did you take? I know this is maybe what.
Yeah, for some of the tasks, let's say sweet, where the classes were well represented, we didn't have so much unbalance. Okay.
Yeah.
For the others, the less represented one, yes. And even then, now we used the same approach for all the modeling tasks. But this is another thing for the future. Let's say. What I like when there is unbalancing is to do undersampling. Okay, I prefer that to other kind of approaches. So, this is another thing that we can do for the future. But of course, there are some tasks where things are okay because they are well represented, as Christian said. Like the sweet class or the bitter class. So this is something that you can do when you model less represented ones. Yeah.
Thank you.
Yeah, you're welcome.
We have another question in the chat, two actually. I was just wondering, can these predictions also predict detection threshold ranges? And I guess you kind of answered that, but
I don't know, Christian, do you want to reply or I reply?
Yeah, better you, David. I don't understand very well the question.
Me too. But I think it's
can you predict concentration at which you can detect that sweetness?
No. This is not
the threshold value.
Detection threshold ranges. No, you can predict something that have been used to train the models. So this must be available as let's say experimental information in the database. And I think this is something that we don't have as an information. Yeah, I mean the models so far we did are qualitative, so they can predict a qualitative label for each chemical. Okay, as I said, as sweet, non sweet, bitter, non bitter, and so on.
Okay, Roberto has a question. Have you performed an importance analysis to assess what are the chemical features that contribute most to defining the different classes?
Yes, we did it because when you do QSPR, then the nice thing is to do, let's say, try to do mechanicistic interpretation on the basis of the descriptors. So yes, we did it, and tried to justify why some of the descriptors were selected in the modeling, specifically for the classification task we were modeling. So yes, you have then the information in the publication, but you can do it because you can use the coefficients of the model, for example, or when you do random forest, you can use the variable importance to assess it. And this is something important when you do, especially QSPR QSAR for toxicological property, but even in this framework, it's important to try to let's say, give a sense to the features that you select.
And there's a question, there's two comments from LZX, which is also called Manti. Whether you have benchmarked the classical machine learning methods versus modern deep learning approaches like graph neural networks and ChemBird, and below, if not, do you think that those models could improve predictions, especially for the small classes, umami and sour and those?
Well, this is an idea for the future, but not to improve models. I think models have already very nice accuracy, and of course, these models cannot be perfect because you have a kind of error you have to accept. So more than for improving the models, we are thinking to use neural networks to try to do a multitask classification, because as Christian said, you can have for each molecule more labels. So that's another thing that we are going to try. So far, regarding the modeling, so far we used, let's say, the classical methods, because we got enough, let's say, accurate predictions, let's say.
In addition, in the ChemTaste predictor paper, there is a comparison of our models with respect to the best models published in the literature. In some cases, our classical QSAR models are better than more advanced classifiers, for example, neural networks. Yeah. So keeping the model as simple as possible is the best option, in our opinion.
Yeah, makes sense. And then there's a question from Sarah. Can this model help with molecules that have bitterness and sweetness sites together?
Not these ones, because these are independent models. Of course, you can combine them, so you can get a new chemical and you can predict with each of these independent models, and then you can combine the predictions. Okay, so if the sweet not sweet and the bitter not bitter models, they both predict as bitter and sweet, then maybe there is a situation like this one. Another option is in the future to have a model which is also able to, as I said, to model all these tasks at the same time. So to be able to handle these multi-labels, okay? I don't know how to call it. Yeah, it's I call it multitask modeling. I don't know. I mean, but the idea is you can predict something as bitter and sweet at the same time, because some of the molecules are actually labeled in this way.
Yeah, makes sense.